Hi everyone, are you interested in learning something new?
To gain more knowledge,
How does it work? And to know its importance.
So before that, let us introduce ourselves. I am Harley Sadnani.
I am Ayishia Detal.
I am Ysha Athena Palce.
I am Velasco.
And I am Jessie Dios.
And we are the Group 9.
And I have a question for you. What do you think is the role of protein in metabolism?
So, protein metabolism is really important process in our bodies.
It's all about breaking down and building proteins.
When we eat proteins, our body breaks it down to smaller parts called amino acids.
These amino acids have different jobs, like giving us energy or helping us make new proteins.
Protein metabolism helps make our muscles strong, fix any damaged tissues, and grow rapidly.
Do you want to learn more?
Let's dive into our main discussion, which are Urea Cycle,
The cycle of biochemical reaction that produces urea from ammonia.
Amino acid carbon skeleton, which is the carbon skeleton, refers to the body structure of the molecules.
Amino acid biosynthesis. The biosynthesis of amino acids involves several biochemical pathways in which amino acids are assembled from other precursors.
Hi everyone! I thought it would be interesting to discuss urea cycle today. But first, what is urea? Urea is a substance our body makes when it breaks down protein. It's a key part of urine and helps our body get rid of extra nitrogen. The liver produces urea as part of waste removal process, and the kidney then excrete it in the urine. The body uses the urea cycle to transform toxic ammonia into urea. It is a series of biochemical reactions in the liver that helps eliminate excess nitrogen from the body. It converts ammonia, a toxic byproduct of protein metabolism, into urea, which is then excreted in urine. The gut flora naturally produces ammonia as well.
Here are the 4 steps for the urea cycle. First step, carbamide transfer group is the carbamide transfer group is associated with the transfer of carbamyl group from carbamyl phosphate to ornithine forming citrulline. This reaction is catalyzed by the enzyme ornithine transcarbamylase. The urea cycle is a series of biochemical reactions that occur in the liver and some other tissues, playing a key role in the disposal of excess nitrogen generated during amino acid metabolism. The carbamyl group is initially formed by the combination of ammonia and bicarbonate with ATP to produce carbamyl phosphate, a reaction catalyzed by the enzyme carbamyl phosphate synthetase. The carbamyl group is then transferred to ornithine in the reaction involving ornithine transcarbamylase, resulting in the formation of citrulline.
Part of the urea cycle takes place in the cytosol, and part of the cycle takes place in the mitochondrial matrix. The bicarbonate reacts with ammonia to form carbamyl phosphate. Reaction of ornithine with carbamyl phosphate gives rise to citrulline, which moves from the mitochondrial matrix to the cytosol. Aspartate reacts with citrulline in the cytosol, producing argininosuccinate. Fumarate is cleaved from argininosuccinate, giving arginine. Urea is hydrolyzed from arginine by arginase and excreted, leaving ornithine. When ornithine moves from the cytosol to the mitochondrial matrix, the cycle can repeat, converting more ammonia and bicarbonate to urea.
Second step, citrulline and aspartate condensation. Citrulline and aspartate come together in a condensation reaction, catalyzed by the enzyme argininosuccinate synthetase. This enzyme facilitates the formation of argininosuccinate by linking the amino acid citrulline with aspartate. The reaction involves the transfer of an amino group from aspartate to citrulline, resulting in the formation of argininosuccinate. The overall reaction can be represented as follows. Citrulline plus aspartate will form argininosuccinate. The condensation results in the formation of argininosuccinate,
a key intermediate in the urea cycle. Citrulline plus aspartate, argininosuccinate. This step is crucial in the urea cycle because it contributes to the synthesis of arginine, another amino acid. Arginine is then further processed in the cycle to produce urea, which is excreted from the body. The urea cycle is essential for maintaining nitrogen balance and preventing the buildup of toxic ammonia in the bloodstream.
The third step is arginine-succinate cleavage. The cleavage of arginine-succinate is a significant step in the urea cycle, and it is catalyzed by the enzyme arginine-succinate lyase. This enzyme breaks down arginine-succinate into two molecules, which is arginine and fumarate. So, arginine, this amino acid, is released during the cleavage of arginine-succinate, while fumarate is the other product of the cleavage is fumarate, a compound in the citric and acid cycle. The overall reaction can be represented as follows. Arginine plus fumarate is equal to the results of arginine-succinate. So, the released arginine is further processed in the urea cycle, ultimately leading to the formation of urea, which is excreted from the body. This entire cycle is essential for eliminating excess nitrogen, primarily in the form of ammonia produced during the breakdown of proteins.
Last step, urea from arginine hydrolysis. Arginine hydrolysis is a biochemical reaction in which the amino acid arginine is broken down into constituent part through the addition of water. The enzyme responsible for catalyzing this reaction is called arginase. The hydrolysis of arginine results in the formation of urea unitin plus H2O results to urea plus orthinin. The released urea is eventually excreted in urine, while orthinin participates in further reaction within the urea cycle.
Basically, urea cycle is crucial for maintaining metabolic balance by detoxifying ammonia, a toxic byproduct of protein metabolism, and converting it into urea for safe excretion. This process prevents hyperammonemia and neurologia damage. The cycle also contributes to nitrogen balance, aids in amino acid metabolism, and indirectly supports energy production. Sustaining the functionality of the urea cycle is vital for overall metabolic health and the prevention of disorders.
Let's move to our next topic, which is amino acid carbon skeleton. Amino acid carbon skeletons are like the building blocks left after we take a weight apart called the amino group. These leftovers are important in our body energy production, making sugar and creating fats.
It refers to the core structure of an amino acid, excluding the amino acid group, NH2, and the carboxyl group, COOH. It consists of a central carbon atom bonded to a hydrogen atom, a carboxyl group, an amino group, and a variable side chain R group that distinguishes each amino acid.
Let's proceed to its function. The carbon skeleton of amino acids serves various functions in the body. After the removal of the amino group through deamination, the carbon skeleton can be utilized for energy production through different metabolic pathways. It can enter the citric acid cycle or Krebs cycle to generate ATP, be converted into glucose through gluconeogenesis, or contribute to the synthesis of fatty acids.
The carbon skeleton in organic molecules provides the structural framework, determining the molecule shape and properties. Variation in carbon skeleton arrangement contribute to the diversity of organic compounds, influencing their functions, such as serving as energy sources, building cell structures, or participating in biochemical reactions.
Glucogenok amino acid. Glucogenok amino acid are amino acid that can be converted into glucose through various metabolic pathways. During catabolism, These amino acids generate intermediates that can intergluconeogenesis pathways, leading to synthesis of glucose. This is important for maintaining blood glucose levels, especially during fasting or periods of increased energy demand.
Example of glyconic amino acid includes alanine, serine, glycine, cysteine, histidine, arginine, proline, threonine, valine, methionine, isoleucine, and tyrosine. Their carbon skeleton can be transformed into molecules such as provate, oxalic acid, or intermediates in the citric acid cycle, which can be then converted into glucose.
Ketogenic amino acid. Ketogenic amino acids are amino acids that can be converted into ketone bodies, which are molecules produced during the breakdowns of fats. Two amino acids, leucine and lysine, are considered exclusively ketogenic, meaning their metabolism leads to production of ketone bodies.
Last is amino acid biosynthesis. In this topic, we will understand the difference between essential amino acid and non-essential amino acid in terms of their biosynthesis pathways.
Amino acid biosynthesis is the biological process through which living organisms synthesize amino acids, the building blocks of proteins, from simpler molecules. This intricate process involves enzymatic reactions that convert precursor molecules into the diverse set of amino acids essential for various cellular functions.
Here are the functions of amino acid biosynthesis. Amino acids have served crucial function in living organisms.
Protein synthesis. Amino acids are the fundamental components of proteins. During protein synthesis, ribosomes link amino acids together based on the information encoded in DNA, forming polypeptide chains.
Next is enzyme activity. Many enzymes which catalyze biochemical reactions are proteins composed of amino acids. The specific sequence and arrangement of amino acid determine an enzyme's function.
Structural support. Proteins provide structural support to cells and tissues. For example, collagen, a protein made of amino acid, is essential for structure of skin, tendons, and bones.
Transport and storage. Some amino acids play a role in transporting molecules across cell membranes, while others are involved in the storage and transport of nutrients within the body.
Immune system function. Antibodies, crucial components of the immune system, are proteins composed of amino acids. They recognize and neutralize foreign substances in the body.
These diverse functions highlight the significance of amino acid in maintaining the structure and function of living organisms.
The key distinction between essential and non-essential amino acid lies in the organism's ability to synthesize them. Non-essential amino acids. Biosynthesis. Organism can synthesize non-essential amino acid through their metabolic pathways. Endogenous production. The body can produce these amino acids internally, and there is no strict dietary requirement for them.
Essential amino acids. Limited biosynthesis. Organisms cannot synthesize essential amino acids or cannot produce them in sufficient quantities.
Next is dietary requirement. Essential amino acid must be obtained from the diet since the organism depends on external sources to meet its needs. The biosynthesis pathways for non-essential amino acids typically involve various metabolic reactions within the cell.
In contrast, essential amino acids need to supply through diet because the organism lacks the necessary enzymatic machinery or metabolic pathways to produce them adequately.
It is important to note that the classification of amino acids are essential or non-essential can vary among different organisms. While certain amino acids are essential for humans, they may be non-essential for some bacteria or plants due to variation in their biosynthetic capabilities.
Here's the importance of amino acid biosynthesis. Amino acid biosynthesis is crucial for building proteins, essential for cell structure and function.
Since the body can produce all amino acids, obtaining them through diet is vital. Amino acids also contribute to neurotransmitters, energy production, and immune function, impacting overall health and psychological processes.
In summary, this process, urea cycle,
Amino acid carbon skeleton
And amino acid biosynthesis are like an intricate puzzle, each piece playing a vital role.
As we finish, let's acknowledge the complexity and connection in these biological activities.
And stay curious about the wonders of biochemistry. Ay na.
Anong left?
Baboy na lang. Bye! Bye! Okay na. Yo. Okay, tapos na.